Welcome to this, our sixth session in the series of Reverse Rett's Emerging Gene Therapy Bite-Sized Sessions. I'm Catherine Sim, and I'm the Operations Manager at Reverse Rett, and also parent to Georgina, who has Rett syndrome, and Georgina is 28 years old. So like you all, parents of people with Rett syndrome around the world, at Reverse Rett, we're eagerly awaiting more information from the ongoing gene therapy trials, which are currently taking place in Canada and the USA. We're also waiting to hear the start of the UK trials, which are projected to start this year. We know how keen families are to learn as much as they can about emerging gene therapies and to better understand what we can all expect moving forwards. That's why we've planned these bite-sized sessions so that we can share up-to-date information with you and hear from experts in the field in the format where you can ask questions directly of the speakers. We try to keep the sessions bite-sized, which means we start on time and finish on time, answering as many questions as possible. We are recording today's session, so we do ask that you do not make or disseminate any of your own recordings. We will share the recordings with you by email after the session today. Please keep yourself on mute throughout the session so that the sound quality is as good as possible. Can I now ask you all to flip on your cameras briefly for a few minutes for security reasons? We do know that many of you are listening in on the fly, caring or working at the same time. We don't mind what you're doing. We just need to make sure everyone is who they say they are. So hello, Louise. Hi, Abigail. I've already said hello to Neil. Emily, hello. Thank you for joining. Did you get in all right? Well, obviously you're here, so I'm going to assume that that's a yes. Okay, we are still waiting for a couple of you just to pop your cameras on, please.
Oops. Finally.
I do like to see all your lovely faces as well.
Okay.
Right, so lastly, if you do have any questions that you'd like to ask, which you weren't able to send in advance, there is at the bottom of your screen a little chat option. And if you pop your questions in there, what we'll do is we'll collate them all together and then we'll ask them at the end of this session today. So with that said, I'm now going to hand you over to my co-host, Rachel Stevenson. Good afternoon, Rachel.
Hello, Catherine. Thank you. It's great to see everyone here. Thank you very much for joining us today. I'm Rachel Stevenson, CEO of Reverse Rett, and mum to Amber and stepmom to Beth, both of whom are adults with Rett syndrome. At Reverse Rett, we've been working for quite a long time now to accelerate treatments for Rett. We've funded over £8 million worth of clinical and lab research, some of which has led to the gene therapy programs which are currently taking place. And we've also funded the CIPP Rett Centre in London for about 5 years now. So the CIPP Rett Centre is a specialist precision medicine centre, which looks after more than 200 complex patients with Rett syndrome from all around the UK. And they have a particular emphasis on emotional, behavioural and autonomic dysregulation symptoms, which I'm sure Jay will go into, and a strong emphasis on clinical research. And so today's guest, Dr. Jatinder Singh, or Jay as we call him, has worked with Professor Santosh and team for more than a decade. Jay is Precision Medicine Research Manager and a Research Fellow at the Department of Child and Adolescent Psychiatry at the Institute of Psychiatry at King's College London. Jay came to the team from the pharmaceutical sector, where he worked in a variety of different target therapeutic areas and in all different clinical trial phases, doing consultancy and scientific advisory work. To date, Jay has co-authored 10 papers on Rett syndrome, 8 as the first author, which I think means he did all the work. No, I'm just kidding, but basically something like that. Some of Jay's papers have been about using existing rare disease and literature from traumatic brain injury to establish meaningful treatment pathways for managing the symptoms of Rett syndrome. So symptoms that cross over, the EBAD symptoms that cross over from other conditions. As we know from rare diseases, and especially with the newly developed emerging therapies like the gene therapies, much of the time we're treading an unbeaten path, pioneering new treatments. And so that's why we look to the experiences of other rare disease groups and other overall disease groups to learn what we can to help our community moving forward. And to this end, last year, Jay co-authored a new paper, which is entitled Evidence Synthesis of Gene Therapy and Gene Editing from Different Disorders, Implications for Individuals with Rett Syndrome, a Systematic Review, which will be the main focus of the conversation today. But I'd like to start, Jay, by just welcoming you, thanking you very much for your willingness to do this session today, and asking you to tell us a little bit about yourself and how you came to be working where you are. So go ahead.
Sure. So, I mean, I like to start this talk by telling you a bit of a story about myself. So, I mean, as a child, I mean, I was severely impaired by my speech, so I didn't really learn to develop any speech until I was like 10, 11 years old. And then from that point, I mean, I didn't really have any peers, and going to childhood was incredibly, going to school in those times I found it a incredibly tough start. And being from an Asian background, I'm Sikh myself. I mean, like there wasn't any services around, many services that my parents could be referred to, like speech and language and so on and so forth. So, but then after moving past 10 years old, I did learn to develop my speech, and I can speak okay now, but it does have some instances that you'll understand in my talk where my speech does get impaired. But what does that mean to the whole perspective of things? I think losing my speech was a significant outcome in my childhood, and I mean, I understand that it can have a significant impact on my parents and my siblings as well. And I understand from this perspective of patients and the families as well, who many of the Rett patients aren't able to speak themselves. So I'm sure, like myself, I mean, when I wasn't able to speak, I could speak internally inside, and if that's the case for Rett patients, where they have some inside voice that they can communicate, but losing your voice has a significant impact. And I think I have this endearing affiliation with parents and the patients and families and caregivers from Rett syndrome, because a small part of me does understand what you're going, even though it's from a speech, and I've learned how to speak. But I think that's why I'm so endeared to the whole reverse Rett community. And if I can give something back through my clinical research, and I'm also doing the work I do in the CIPP Rett Center, that would be a worthy cause for me.
Well, I really appreciate you sharing that with us, such a personal thing. And
Okay,
you know, it's actually really good to hear you talking about having an inside voice at the point where, so can I ask you, did you lose your speech, or did you just not speak until you were?
No, I mean, so I was effectively non-verbal until I was like 8 years old. So I didn't really gather. Maybe it was my mum used to say you did have a few utterances, but yeah, I think yeah, no, I wasn't really able to speak at all, apart from a few utterances.
I mean, of course, we in the Rett community don't equate a lack of speech with a lack of intelligence, and you've already explained, you know, that you had your inside voice, but you know, I consider you one of the most complicated people ever to speak to, that's in my realm of people that I have to speak to on a daily basis, and that's because of your you know, the complexity of the issues that you work on. And so how do you go from being in that situation where as a, you know, you've got a disability as a young person to then excelling obviously academically and then professionally?
I mean, to answer that, I mean, so many people I've spoken to have mentioned this to me. So where I'm not really good with speaking, I can see words completely differently. I see words as shades and numbers. So as my colleagues have told me, whether I've got some savant-like skills that high functioning autism people do have. So I think that part of my, so during them years where I mean that part of my development which haven't been adapted to speak, I think I have overdeveloped regions of the brain which can help me assimilate large amounts of information. And luckily for me, I was able to find the positions I was able to work in, to use it in my career, from working in the pharmaceutical industry to where you have to assimilate large amounts of data and information, put it into meaningful reports and stuff, and then and then just things. So I think I have got a certain more developed parts that I can see words and huge amounts of data completely differently from other people, which people in clinicians in who I've worked with have told me this, that you do have what is known as savant-like features when it comes to interpretation of words. Because I see a unique feature of me, if anyone has a conversation with me, I can recall anything they say, whether it's a year ago or 5 years ago, but it's not that I recall the words. I can see it in different shades of color, if that makes any sense.
I think it's always difficult, isn't it, to understand how other people's brains work. And it's also a situation where I think a lot of the time we think we're normal because that's our perception, you know, our perception of what's happening to us is typical because that's all we've known. And so, but as I'm often reminded, that's not necessarily by my children, usually that's not necessarily the case in my case. But your job title, just moving to now what you're doing now, your job title is Precision Medicine Research Manager. So I know what you do and the breadth of what you do and how important you are in the CIPP Centre. But could you tell everybody else here what you do on a sort of day-to-day basis?
Yeah, sure. So what I do on a day-to-day basis, so so each patient which comes to the CIPP Rett Centre has to wear a watch-like device to work out their biometrics. So whether they have autonomic dysregulation. I mean, if Rett patients have severe autonomic dysregulation, do not really respond to treatment and so on and so forth. So all patients have a watch device and also have a pharmacogenomics test done. Basically, what pharmacogenomics is how your genes respond to the medications you take. So if you're taking sertraline or paracetamol, ibuprofen, any painkiller is doing that. So when they have a pharmacogenomics done, we get sent a pharmacogenomics report from the lab, and I interpret that report. And alongside Professor Santosh, we give directions to the clinical team. Any patients, this is also include all the patients in the not only in the CIPP Rett Centre, but all the patients, rare and non-rare, how they respond to medications. Also look at the watch data, and when Professor Santosh describes it to patients, what they how to watch data, whether they've got autonomic dysregulation. I also do precision medicine reports. Now, basically what precision medicine reports are, their report, which takes account of all the data that the patient has. So it could be watch data, the past medical history, the different types of medications beyond. But the key issues, the two main factors for these patients, they're not responding to any type of treatment, and they become treatment non-responders. So I get given those patients to see, by doing a deep dive in all their information, pharmacogenomics watch, can I find any patterns in their profile which can turn them into treatment non-responders to treatment responders? And I feed that back into the clinical team and Professor Santosh. Aside from that, I also have oversight of the CIPP Rett Center. So that's working really closely with Professor Santosh to see if any clinical trials are coming into the team, any gene therapy trials, trying to organize them, anything to do with the CIPP Rett Center, the logistics behind it, dealing with the clinicians who work in the center. And as a side role, I try and do my clinical research, but unfortunately, I don't get any time to do my day-to-day. So all the papers that Rachel mentioned in the beginning, unfortunately, they're done and out of hours and mostly in the weekends. But it's I think it's important to do, because it's good to give to write these papers, to give something back to the overall Rett community and also the rare disease community as well. So that's what I basically do. But I think the part of the CIPP Rett Center does change a lot. So like you could have a rigid way to do things, but on a day-to-day, it could all of a suddenly change. Like when a parent comes in and they don't have access to this, or they need this, and so I'm involved with that with other team members to ensure that the patient journey is as good as it can be when they come into the CIPP Rett Center.
Thank you. I think that covers it really well. Just before we go to the next question, I think it would be helpful if you just summarize autonomic dysregulation and how it's fundamental in Rett syndrome.
Yeah,
Because we've mentioned it a few times.
Yeah.
Yeah.
So what autonomic dysregulation is, just imagine you have a seesaw, and the seesaw is your autonomic system. On one hand, you've got the sympathetic autonomic system. On the other hand, you've got your parasympathetic system. And basically what autonomic is dysregulation if the seesaw tilts that way or tilts that way. For example, me in my giving this talk now, probably my autonomic dysregulation is slightly skewed to the sympathetic, means that I'm having my hands have got like wet with sweat and stuff like that. But what usually happens in Rett patients or anyone who's got autonomic dysregulation, it just shifts like this all the time. And it's that autonomic dysregulation occurs, it can have a knock-on effect of the emotional components and the behavioral components. So the emotional components are like anxiety, phobias, mood dysregulation, and the behavioral components can be repetitive behaviors, stereotypies getting worse in Rett patients, agitation. And if you combine the behavioral and the emotional with the seesaw going like that, which is autonomic dysregulation, it's known as EBAD, emotional, behavioral, and autonomic dysregulation. And EBAD typically is seen in many patients with rare disease, including Rett syndrome. And a key feature of that is patients with EBAD do not respond to treatment, become treatment non-responders. So one of my roles is to try and tackle EBAD, and the only way you can tackle that is by targeting the autonomic dysregulation, because we find it quite difficult to target the behavioral and emotional. I think it's more easily to target the autonomic dysregulation, so we can prevent or manage EBAD, so we can get patients who are treatment resistant to become responding to treatment.
Thank you. I think, and as well, we should say that when we think about EBAD and Rett syndrome, a lot of the time we think about breathing, but it's kind of before I had language for what autonomic dysregulation was, and my daughter's always been a terrible breath holder, and when she was younger, hyperventilator. And I always used to describe it as being very chicken and egg. So is it a product of the autonomic dysregulation that someone has the breathing problem, or is it that they breathe erratically and then it creates that they're bringing themselves into stuff like flight or fight? Do you see what I'm saying?
Yeah.
Like, because a lot, and everybody knows now, if you're into these things, that it's quite cool to be able to regulate your own breathing or to dampen down the response that you're having by controlling your breathing. So it's a shame we can't harness that for them to just bring them down, regulate their whole situation. So what, I don't know if that's really a question. I suppose the question is, yeah, is it a chip? Is it?
Yeah. So when it comes to breathing dysregulation in Rett patients, there's a part of your central nervous system just above your head, which is called a brain stem. And in the brain stem, you have these bodies. They call them collicular fuse, but basically what that is the central autonomic network, and that's your breathing center. In Rett patients, that hasn't been developed that well, and there's some evidence that they have an immature breathing center in Rett syndrome. And so what that means is that has an impact on the whole autonomic cycle. So if a Rett patient has anxiety, the emotional component, their breathing will get worse. If there's anything, so it all feeds in. So if there's behavioral component, their breathing can get worse. But because the fundamental breathing network is impaired in Rett syndrome, it just has a bidirectional effect, because the autonomic nervous system works both ways. It works in the central and also the peripheral nervous system. So it works both ways. So one can argue that if the breathing network was so-called fine in Rett patients, then autonomic dysregulation should be able to control it. But in most instances it does, and hence why we use buspirone to manage the breathing dysregulation in Rett patients, because that center I talked about, the collicular fuse, is really high density of these receptors called 5-HT, and what locks onto them is a buspirone, and it helps with the breathing in Rett patients. And also by tackling the breathing, you can also potentially help with the emotional experts, such as agitation and anxiety. And as you well aware, that a lot of the antidepressants, which are 5-HT compounds like sertraline and fluoxetine, do work on for anxiety disorders.
Thank you for answering that question. That's really interesting. Okay. The next thing I wanted to ask you was, you have authored a number of papers on Rett syndrome now. And I think many of them, and I think what we should do after this session is we should put the list of some of the different papers that you've authored onto the email that we send out to people, so people can check those out themselves.
Yeah.
There's been a lot that I think have really meaningful suggestions for how people, things that people can take out of them in the view of things that they can recommend to their own doctors, whether or not they're under the CIPP center or not. But what I would like to know from you is, when you've authored all of these papers, what was your favorite paper that you've authored, and why?
Yeah, so me, I think it was the first paper where we put the hypothesis of EBAD in Rett syndrome, and although EBAD, although in some other disorders, rare disorders, they spoke about the behavioral, the B components, and the E, and also there's a lot of information in Rett syndrome about autonomic dysregulation going back to the 1990s. So we put it all together, came up with this construct, which is known as EBAD, because we identified that many of the patients who we have emotional behavior and autonomic dysregulation do have become treatment non-responders. And the reason why the paper was so important, because it describes that how we looked at all the literature out where the studies have done in EBAD, and we came to the conclusion that by targeting the autonomic components will make a good target site for getting a handle on what EBAD is. And because previously people have attempted to look at the E side, which is emotion, and the B side, and they haven't been able to look at combine as holistically what EBAD is, but which we did, and then although that was a hypothesis paper, we have shown that it does work in using empirical data, using the watch data, and also using buspirone, and also like some other medications, that we can manage EBAD. So now we routinely use medication, the CIPP to manage EBAD in these patients.
Also, I think that we should say as well that for EBAD, so you've got the emotional sort of anxiety type things, you've got behavioral, this kind of agitation, even can be violent sometimes, or self-harm. And then the autonomic, which everyone immediately, as we've already done, thinks breathing. But there's other aspects of autonomic dysregulation as well, aren't there? Like things like temperature regulation and
I mean, so with autonomic dysregulation, you also talk about gastrointestinal function, because the autonomic system, we have a nerve called a vagal nerve, which goes all the way down from your spine and all the way down to intestinal system. So when your autonomic dysregulation gets worse, you do have an impact on the gastrointestinal systems. EBAD also covers the cardiac issues. So as you know, some patients have long QT, and the more high risk patients, such as those who've got epilepsy, long QT, and breathing dysregulation, they'll be at more high risk of EBAD, especially the autonomic components. So those patients, we have to take more care.
Jay, sorry, can I just ask, in relation to that autoimmune function, do hormones play a big part in unbalancing that?
Yes, of course. So one of the triggers of EBAD is puberty and stress as well. So there is some evidence out there that Rett syndrome can be classified as an endocrinology pathology. So there is this regulation of hormones as well. Yes, so puberty and also hormone imbalance has a huge impact on EBAD.
I mean, I'm trying to get an endocrinology referral right now, but I have to say, I feel like for people who have children with Rett syndrome, it's a little bit like respiratory. It's one of these things that's clearly a fundamental issue that it's really difficult to get help with, because historically it's not been kind of seen as that kind of disorder. So yeah, evidence that shows that is playing a big part would be really helpful for people in terms of getting referrals. Okay, well, let me ask you this now. So your gene therapy meta-analysis paper came out in 2023, as we said. Can you explain, before we go into why you wanted to write this paper, what a meta-analysis is?
Yeah, so what meta analysis is, you basically what it is, you look at lots of evidence, and you try and use that evidence to answer research question there is. So for example, if I was going to say, is Porsche 911 the fastest car, then I would look at all the other similar cars to Porsche, and my research hypothesis is yes, I think Porsche 911 is the fastest car, and then I'll compare all the other things, and then I'll come up with an end. I'll come up with themes of why it is the fastest car or not. So basically, in a nutshell, is to use all the data out there to answer your research question using stats to say, statistics to say, to answer your research question. Why do you think this Porsche car is the fastest car on earth?
Okay, so why did you want to write this particular paper?
So the reason I wanted to look at this meta-analysis in Rett syndrome, because I was aware that the gene therapies have started to emerge in Rett syndrome, or started to emerge. And then I also understood that there was a lot of studies done in other disorders in gene therapy, but hardly any in Rett syndrome. So I wanted to look at all the evidence in all the other disorders. And then my research question was, what do the evidence in the other disorders tell me about the success or non-success of gene therapy trials in Rett syndrome? And because all the other disorders have been going on for the last 20 years in adults and children, and these are disorders such as Tay-Sachs disease, Huntington's, tuberous sclerosis, sickle cell, MPS, and so on and so forth. I put all that information together to answer some key questions for the Rett syndrome community. And basically, the key things are, what is the time window for gene therapy? What is the best mode of gene? What is the best transport vehicle in Rett syndrome, and whether it would work or not? And based on the information I saw, I tried to come up with some answers. So that's why I did it. The answers I came up with, I put it in a small table at the end, because I know it'd be vital for the Rett community to read the paper. So I summarized all the paper into the small table at the end, where I described the other disorders, the key outcomes from them, and what it would mean in terms of Rett syndrome.
Before we move to that part, though, can we talk about how you decide? So obviously there's a breadth of information. So even aside from this specific paper, how do you kind of whittle it down? So you first of all, you scan the whole of the literature, which is, thousands and thousands of things, and you use specific search terms. I mean, in a way, isn't it a bit like, when you go on Amazon and you want to buy something and it says it's got 9 million reviews, and then there's one little summary at the top now with AI that says, and based on that, this is what most people think. But as we all know, what most people think isn't that scientific. So with something like this, it isn't just a question of explain how it's different to like doing an AI thing and going, AI, tell me if there's any point in doing gene therapy in Rett syndrome, and then it would come back
sure,
mishmash of kind of Frankenstein's monster answer of
sure. So there's two key points. The first point is that when you do any meta-analysis, you have to be guided by the standardized guidelines they have. And basically these are guidelines which tell you how to do it. And anyone, if you do a meta-analysis in socks, what color socks or anything in autism or like peaches or something, you'd have to use these same guidelines. And when you submit it to the journal articles, they want you to use these guidelines, and it's more robust also for two people to independently do it. So if for example, Andy wanted to say, why do his socks disappear each morning? Me and Rachel will do a research. Why does Andy's socks disappear? And I'll do my search, and Rachel will do her search. And that leads you on to my second point. If I was just searching for socks on PubMed, or anything, I'll come up with thousands and thousands of hits. But if I was searching the disappearance of socks, particularly red socks, I'll have more. It'll condense my searches. And going back to how I did it for the Rett paper, I was guided by that standardized framework I did, but my searches were looking at disorders which involving the brain. So to do my searches, and I use Angelman's tuberous sclerosis disorder and Tay-Sachs and Huntington's, although they're completely different to Rett syndrome. But if I search for Rett syndrome and gene therapy, I wouldn't be able to do it because no information exists on it. So, and I think it's just tweaking your terms. I think that's the issue in psychiatry. So if I wanted to do it, I've done a meta-analysis on autism, but if you look at the field of autism, it's such a huge field. It's more attractive that, I mean, people perceive it to be attractive. I'm not saying it is, but I think a lot of people, there's so much literature around the subject, the frequency rate of hundreds of articles published on each week. So doing autism systematic review is also challenging. I mean, and also not many people do meta-analysis systematic reviews because they take a lot of time and they're incredibly difficult to do. But I think part of my neurodiverse component I have during my upbringing and how my brain developed, I find them quite not easy, but I can do them. So I think yeah, so that's, I'm sure I've answered it. I'm not sure if I answered it. Tell me if I'm rambling on, Rachel.
No, you're not rambling on at all. I think it's important that people understand what the point of doing a paper like this is and how it actually does add something. Because I think one of the problems we've had for a long time in the Rett community is that people were trying things like buspirone and, but there wasn't a lot of literature about how this had worked in bigger groups of people, and so to have papers that summarize, okay, this is where we're up to now, is really helpful. And then of course with the gene therapy, I think we get questions every day from people all around the world asking how we think things will pan out. And I think obviously we don't know until these clinical trials happen, but we can, as you say, look to other disease groups. And so and find out what overall happened in them, and it might give us some insight into ways forward. So what were your overall conclusions?
So what I found in the papers, the overall conclusions is to have effective gene therapy, there's a couple of things you have to consider. That's the time window when you provide the intervention, the biodistribution, which means how you give the gene therapy compound, and where is the field going in a couple of years' time. So these are the three main things that came across, and how what it means for Rett syndrome. Yeah, so those are the main things I spoke about.
And I was reassured to see that on page 28, which is after you've put the lovely table 2, where you break down those other conditions, that you have actually said, obviously, I think everybody knows that if you can try and treat the underlying cause of any disorder before the person becomes symptomatic, it stands to reason that that's likely to be more beneficial. And I think the thing that people always want to know in Rett syndrome is, would you consider it that later on it wouldn't be helpful at all. And obviously we've got some evidence from animal models that speaks to that as well. But I've noted as well that on your paper you've said gene therapy or gene editing may still benefit past the plateau stage for clinical stabilization, such as preventing motor decline from worsening. And I think that's really important because, you know, the issue that we see when we look to other disorders more anecdotally, you know, not in the same way that you've done here, but say things that we see in the news where NICE's approved gene therapy treatment for something like metachromatic leukodystrophy, name of it, or and they've only done it for patients of a certain age and not you know, the older patients. And I think that makes people in our community feel quite nervous. And I think it's important to just highlight, you know, and make sure that as we move forward towards gene therapy trials, that we make sure that we emphasize towards gene therapies, you know, going to HTAs, health technology assessments. We make sure that we're emphasizing that sometimes there's differences in these conditions, and there's no, you know, for example, although people with Rett syndrome have a progressive disorder, they don't have a degenerative disorder. And, you know, some of these diseases are fatal, and Rett syndrome technically is fatal, although of course we have devastating situations. In and of itself, for example, in metachromatic leukodystrophy, children typically die between the ages of 5 and 8. And SMA is similar. And so I suppose from our point of view as a patient group, we're always trying to make the distinction between our patient population and these other patient populations where, you know, the deterioration is there in a different kind of way. It's brain deterioration. The other question that I had, and then I'm going to be quiet because there's a lot of questions in the chat, which is really good, is I know when you've talked about Huntington's disease in here, so we've got a list of things that are really childhood diseases, and then, you know, where it's fatal quite early on. And then we've got Huntington's disease, which is awful. But there's not kind of a mention of early intervention there. And I wondered, kind of, you know, because that's something that develops most of the time, isn't it, in middle age? Is that why? I mean, and then could you look at a situation where you thought, well, you know, maybe this could be picked up earlier and people could be given these kinds of treatments earlier, or is it?
Yeah, no, like I think from the with Huntington's is a triplet expansion disease. So when your triplets get to the finite limit, then you suddenly develop symptoms when you're probably in your late 30s and 40s. The reason I put in Huntington's in the paper to emphasize the point about distribution. And that's an important point because in Huntington's, they injected the molecule. It was an antisense oligonucleotide, which is a compound, which is basically, if you have, so the Huntington is a mutated protein, and an antisense tries to unmutate that protein, an antisense. So they gave it intrathecally, and then they wanted to get it into the brain. But when they analyzed the cerebrospinal fluid of Huntington patients, while the mutated protein, which is Huntington's, was decreased in the cerebrospinal fluid of patients, there was no change whatsoever in outcomes in the course of the disorder. And this is the fundamental point, that just because you inject something intrathecally, it doesn't mean it's going to get to the brain. And as you know, in 2021, which is available, this information is available. The reason why Novartis mothballed their gene therapy syndrome in Rett syndrome is because while the preclinical animal models suggested that it was fantastic, when they did the biodistribution studies in the monkey models, they had the same effect. There wasn't any real effectiveness in those monkey models when they injected in the CSF. So one of the key points I'm trying to make using Huntington's and the Novartis failed trial, that biodistribution is an important thing to consider when it comes to gene therapy, aside from all the other things about the MECP2 being over or underactivated and so on and so forth.
I mean, I think I would really like to ask you as a next question.
Yeah, sure. You're
I'm looking at Catherine's
just
face.
feel free to ask me anything.
Well, I'm not going to ask it, but I think that at a later date, maybe we need to come back and ask this question, and then how things, whether or not you could explain how things have developed from the Novartis Avexis program to the Taysha and Nora-G programs, and how those are different, and why it is that we're seeing some early efficacy from.
Yeah, I think the main difference is potentially used to the vector they were using. I mean, I think it does boil down to the vector. So a lot of these, so basically a vector is you need to transport the gene into your nerve cells, and by doing that is an adenovirus 9 vector. It's just they use it 9 because it happens to get into your nerve cells very easily. It's a virus. Adenovirus is a commonly occurring virus that we get, children get, or we get, but this virus does infect nerve cells, and adenovirus 9 has been edited by scientists to make it more easily to get into cells. Now, if you look at the Taysha one, they're both using adenovirus 9 as the gene one. The key difference is that the Taysha one is using a small version of MECP2, and the Nora gene is using the full length of MECP2. They both in theory will work, because the small one, which is based on an MI-rare technology, which is developed in Boston's Children's Hospital, it's been well known in a paper which came out a couple of years ago, that a small truncated form of MECP2 does fully recapitulate all the core features of Rett syndrome, and this is the Taysha thing. And then in the Nora gene one, it uses a different technology. It's full length, but it uses exact, and so the Taysha uses me-rare technology, and the Nora gene uses exact technology. And not to get bogged down what they mean, basically the technology is to stop the MECP2 to under or over amplified. MECP2 is a dosage sensitive gene, so if you have more than 2 copies of it, you get MECP2 duplication syndrome, which is, you don't want that to happen, and it just prevents that from happening. And one of the key differences besides Taysha's intrathecal, and the Nora gene is intracerebroventricular, which they inject inside your brain. And one of the gene therapy studies, when they looked at Tay-Sachs disorder, which is a neurodegenerative disorder, they found by injecting the gene therapy into the thalamus region of the brain, it didn't reach all parts of the brain. So they decided that a combined intrathecal intracerebroventricular may be more efficient in Tay-Sachs. So it doesn't mean that if you give intracerebroventricular, it's going to be better than the intrathecal one that Taysha is doing. You just have to get the gene copy in the right parts of the brain, because as you know, is an X-linked disorder, so different parts of the brain will have some healthy MECP2 cells or non-healthy, and they spread across. I hope that understands.
That's very helpful. Thank you. Right now,
Okay.
Jay, we have officially got to let Catherine ask.
Okay, sure.
Let me back in. I'm like, time. Okay, so we've got quite a few questions, and I've actually really been enjoying watching them coming up and going, oh yeah, I want to know the answer to that. So the first one, I'm sorry, Google Pix 7. I don't know what your real name is, but you have asked this question. Your daughter is 29 with late onset Rett. What are the chances of her being considered for general therapy trials, as current trials all appear to be for children?
Yeah, I think that's a really important thing. I mean, from my point of view, I think the whole age range should be considered for the whole gene therapy trial for children or the adults. I think it's important for the older patient to be considered, not only to clinically stabilize the disorder, but many patients have severe symptoms like epilepsy and breathing dysregulation, which can occur across the entire lifespan. I think it depends what the companies would want to do. Obviously, they want to do the children first because maybe that's the interest in her. I do think it'd be up to us as a Rett community to put pressure on these companies to make sure that the full gamut and spectrum. And I know Rachel and the team, John and Andy and Catherine, would do the level best that all patients are included. And if you need me to help with that, I'm fully 100% on board with that, as I mentioned in my paper.
Yeah, we'll take you up on that, Jay. Thank you. And I know that Rachel and Andy and John and I continue to advocate at every opportunity to make sure that everybody, male or female, regardless of age, will be included in any upcoming trials. That's what we do. It's our job.
Also, I think it's important to say as well that we have had a commitment from both companies that they're wanting to develop these products for people across all age groups who have Rett syndrome, and they've made that commitment to us, honestly.
They have. Okay, the next question is from Jennifer. And funnily enough, we were talking about this very thing this morning. Is the CIPP Rett Centre just for UK residents?
The thing is, so we have a couple of patients come into the CIPP Rett Centre, but the logistics are quite complicated because they need to get the funding sorted out before they come. And so we do have certain instances that we do. There's one at least coming up in June, and I know that there was a lot of logistics which the funding had to be done at their thing to get sorted out and to seen. So yeah, it does, but it's not an easy thing to do. But it
Do you
can
think
be
in
done.
the first instance they should actually just write into Professor Santosh?
Yes. I think usually
him?
what you should do, just write to the CIPP, the CPMRS email inbox, and we will action it. It always comes through the MDT Wednesday meeting. If there's a new patient coming in, we discuss it at referral, and Professor Santosh and the wider clinical team, we discuss it then. And then we also have other sessions of the CIPP Rett Centre team where we discuss it further.
Brilliant. Okay, thank you. Google Pixel number 7 again has asked, so his daughter has a VNS, a vagus nerve stimulator implanted, and can that adversely affect any medications such as buspirone?
Yeah, I think the best thing to do that is to seek advice from the doctor. I think he'd be in the best position to answer that. I know in usual instances, the vagal nerve stimulating devices are used quite a lot in other fields, but Rett is not a typical case, so I would defer that to the key specialists who are involved in that.
Brilliant, thank you. Abigail says that her daughter doesn't suffer from EBAD, so she would be concerned to give antidepressant, anti-anxiety type medication. And what alternatives would you typically consider for significant breathing irregularities that aren't in that group of medicine?
Yeah, so I think for the breathing issues, I think so we predominantly give buspirone to manage the breathing issues, and if it just happens you do have anxiety, it won't have a direct impact on anxiety, but by helping managing your breathing, if the patient does anxiety, it will help in that way. If the patient doesn't have anxiety, I think buspirone does have is effective to manage their anxiety, regardless you have EBAD, because although EBAD is a collective term, you could have so if you look at the autonomic components of EBAD, if you just look at the autonomic, that involves gastrointestinal, cardiac, the breathing dysregulation. That should not stop you from taking any type of medication for the cardiac, for the breathing, regardless if you have EBAD or not. EBAD is just a collective term for the whole gamut of symptoms.
Thank you. For everybody in the chat, just to let you know that Wesley has just popped the email address for the CIPP Rett Centre into the comments. So if you want to make a note of that, that is in there. Okay, so Louise asked, I was going to ask this as I believe that gene therapy treatment for SMA type 2 is only available to children under the age of 2. And that made her nervous about what gene therapy might look like in Rett syndrome and what age it may be available for or most effective. So is there a golden window? Is there a golden time when actually it's going to have the biggest impact?
I think the gene therapy approval for SMA is based on a really strict criteria that the NICE have formulated. I think a couple of things it depends on. It depends how number of mutated copy variants you have of the SMA. If your child has an X amount, then they get triaged into the gene therapy. So it does depend on copy number variants, and their criteria is quite rigid, and I don't think that's transferable to Rett syndrome at all, because spinal muscular atrophy is a more focal target, and they've made clear guidelines. I think Rett syndrome would be different in this case, and yeah, I think it would be different in this case.
I also think it's important to say that, you know, again, it's a degenerative condition that's usually fatal for children, and with our condition, you know, we've seen lots and lots of replicated studies in mice that show that function is restored in mice that have even reached late stage disease. And so it seems to be more a case of the protein is missing. You take this protein away and there's a problem, and you put it back and the problems are diminished. And so what you have to really worry about is there are some things that are entrenched with someone like my daughter, who hasn't walked since she was 17, which is now 11 years ago. And will she be able to jump back up? No, but it may be, I think this is what Jay's paper was saying as well, it may be that she could be clinically stabilized. So some of the other problems that she has, and I think this is on the table as well, which, by the way, don't feel like you can't get embedded in these papers and just properly look at what's written in here. It could stabilize patients. This is in panel A now on page 29. Prevent symptom progressions such as dystonia, spasticity, Parkinsonian-like tremor, rigidity from worsening. Those are big things. And the role of the patient group is to be able to say, listen, powers that be, this is the difference to the quality of life, and to, you know, adding into the whole benefits and how this impacts the person, the family, and the wider society, what the differences are that can be made by getting rid of some of these problems, what the cost savings that can be made by getting rid of some of these problems. And you can bet you know, anything you like in the whole entire world, that we're going to be there making these arguments for our people, because when you splice out Rett syndrome, exactly what the cost, human and otherwise, is of having these different symptoms, and what the benefits are of taking them away or improving them.
Okay, this is the last question because we're 2 minutes over already. So Sergi has asked, and it's a kind of double-edged question, is there any chance to expand clinical trials to other countries in Europe, and more likely as a kind of knowledge support to other researchers? And if so, are there any countries that have already asked for your help on this? I mean, that's really more to the pharma companies, but I don't know if you have any information, Jay.
No, as far as I know, I mean, I know Eurogene are doing 17 sites across the world, and maybe a couple in the UK. But I think, because of the non-disclosure agreements and the confidentiality clauses, if there's other sites, we're not privy to know what these sites are. But yeah, no, I can't really, unfortunately, I can't answer that, because a lot of the pharma companies like to keep it. But in terms of the other research centers, we haven't been in touch with any other research centers. But one thing I'd like to say, if you need me to come again on this bite-sized meeting, and you just want to have a question and answer session with any of the parents and caregivers, if you want to talk about trifetinide or anything like that, it doesn't have to be about the papers or anything. I'm more than happy to come and do a Q&A session, if that would be helpful.
Yeah, that would be really cool. But I think as well, you know, if we just emphasize to people that we do try and convey the need that's all over the world, and the best way for you to help us with that is by registering and getting people in whichever country you're in to register, so that we can see where the dense Rett populations are. And then the only other thing is to say, I think they usually just pick the big 5 in Europe. So that would be Germany, UK, Spain, Italy. Is it France? I can't remember. Off the top of my head, I would say that's what we go for. But we do have heavy concentrations of people in different countries. For example, we've got quite a few families in Poland that are constantly coming forward. And the more we can emphasize to companies this issue's out there, that will obviously help. But we are really mindful of the fact that everybody is eager to be part of clinical trials, and we are too. You know, I think it's important for us to say as well that we're expecting UK clinical trials for children, but we aren't expecting any for adults, as much as we would love that. And several of us have our own adult children with Rett. But I think the most important thing to remember when we're thinking about the clinical trials is the idea is that the companies are trying to demonstrate that they work and that they're safe in a population where it will be really easy to show that. And then they'll bring forth, then perhaps broaden the trial so that more people are included, or expand the age ranges so that more people are included, so that they can show the payers and the regulators that this works for, you know, across the lifespan. And then hopefully we'll get it rolled out to everyone. But it's all a process. And we keep going.
Okay, so from me, thank you, Jay. It was really, really interesting.
Thank you.
And I hope everybody got from it the same things that I did. It was brilliant. Thank you to everybody out there, all the little black boxes that we look at every time we do one of these bite size. Thank you for coming along to today's session.